bioinformatics was catalyzed by his desire to make an impact on scientific progress without world-class facilities. Specifically, some challenges that Rohith faced were limited access to mentors, as well as limited computing power. Rohith conducted his most recent work at UC San Diego and has been honored as an Intel finalist and will be publishing a future paper as the first author. In addition to being an avid researcher, Rohith does debate, theater, outdoor sports, and basketball. In the future, Rohith hopes to study something related to engineering or math.
All right. So hello, everyone. How does the PowerPoint look? It's, yeah. Cool. Thank you. So I'll just get right into it then. So my project studied this disease called non-alcoholic fatty liver disease. And so first we'll start with some background. So this disease, non-alcoholic fatty liver disease, it actually affects as many as 20% of Americans. And it's a significant risk factor for HCC, which stands for hepatocellular carcinoma, or in other words, liver cancer. So you might be wondering if it affects 20% of Americans, why you haven't really heard of it more. And the reason might be because most of these people who are afflicted with fatty liver disease don't even know they have it, because fatty liver disease in and of itself is not a huge deal and you can just live your life relatively normally. But the reason why it is a significant problem that we want to study is because, as I mentioned, it's a significant risk factor for developing liver cancer. And the reason, what we're really interested in looking at when we study fatty liver disease is not the disease itself, but how the disease might progress to future stages. And currently little is known about the nature of this progression, so that's what I kind of wanted to look into. So as you see from this diagram, the stages you go to are, you start with normal liver, and then whether it be dietary issues or genetic issues, you might progress to steatosis, or in other words, non-alcoholic fatty liver disease. And from there, because a fatty liver is not a happy liver, it might get inflamed, and you might progress to steatohepatitis, which is called NASH, or that's when you have liver inflammation. And once you have that liver inflammation, then what can happen is that your liver gets really annoyed and there's this violent response. And that violent response can be characterized by excessive cell division and other signs that we see in the manifestation of cancer and liver fibrosis. And that's where where there really starts to be significant problems. So the goal of what I wanted to do was, first I wanted to discover markers and mechanisms that facilitate this progression, specifically from a normal liver to non-alcoholic fatty liver disease to then NASH. And then I also wanted to identify, I was also looking at this one specific potential causal factor, which is DNA methylation. And so I'll go into that a little bit now. So DNA methylation is this chemical modification of a DNA strand. And as some of you might know, DNA is what relays messages to your body's cells and gives them instructions on what to do. And so when you chemically alter that DNA strand through DNA methylation, you change the way a DNA strand is able to relay information to the rest of its cell, and as a result, you change how the cell's functions are affected. And so the methylome is what we refer to as the comprising all of your body's DNA methylation signs. And so I wanted to also study how interactions between the methylome and transcriptome might facilitate progression together, because DNA methylation directly affects transcription or gene expression, and in turn directly affects how your body functions. And because people in the past had identified DNA methylation as a potential underlying cause, I wanted to go into it further and explore specific ways that it could affect the progression of fatty liver disease. So one of the things that I think differentiated the way I looked at the disease from the way people in the past have looked at the disease is that I studied genes as from a network-based approach. And the reason I did this is because genes don't work in isolation, they work in teams. And each member of a team has a different role. And in order for the team to perform a function, each member of the team needs to perform its role the way it's supposed to. And so because genes work in teams, I wanted to analyze them as teams. And so I analyzed the data I was looking at, gene expression data and DNA methylation data. I analyzed it by reconstructing data-driven networks. And so the goal of my project would be that the data-driven networks, or the unbiased networks I constructed, basically without inputting any actual biological knowledge into them, hopefully those networks would yield meaningful insights on the actual biology, which is what we'd actually be interested in. So first, I constructed a co-expression network from gene expression data. So gene expression data is essentially you can picture it as someone conducted a study where they had a certain number of patients with a normal liver, a certain number of patients with a fatty liver, and a certain number of patients with an inflamed liver. And so for each gene, they have a vector of gene expression levels for all of those patients. And so by correlating those vectors together, you can construct a network where each node represents a gene, and then the edge connecting each node to every other node represents how related those genes are, per se. And so I constructed that network, and then, as I mentioned, I wanted to not just look at gene expression data, but also integrate it with DNA methylation data to look at how DNA methylation might be affecting gene expression and facilitating progression. So then what I did is, once I had this network, I constructed modules within that network. And a module in a network is essentially a group of nodes, in this case genes, that are highly connected to each other. So there are a lot of edges they're very related to each other. And so in that way, you can define modules within your network of, say, 50 to 100 genes or even more, where those genes are very highly connected within the module, but perhaps not as connected to genes outside the module. And then I looked at how those modules were preserved, or in other words, how the relationships between genes within the module were preserved when you define those relationships based on gene expression data, as opposed to when you define those relationships based on DNA methylation data. And so the way you can think of module preservation is as follows. So imagine you have a set of dots on a sheet of paper, and you draw a random set of lines connecting those dots. And then you take the same set of dots on another sheet of paper, and then you close your eyes, and you try to recreate the same set of lines. Now, maybe there might be some areas on the second sheet of paper that look pretty similar to the first sheet of paper, but it's probably going to be somewhat different. And so by looking at module preservation, you're looking at specific areas of dots or clusters of dots where those lines don't rearrange themselves as much. And that would indicate that that particular group of genes, the relationships are preserved. So I did that in a much more mathematically oriented way. And so what ended up happening was that we identified 6 modules, each of, anywhere between 20 and 100 genes, where the relationships, at least from the data, from the DNA gene expression data we had, were preserved in the DNA methylation data we had. And so that was interesting because gene expression data and DNA methylation data are two dramatically different forms of data, different types of data. So if relationships are very statistically significantly preserved between these two very diverse forms of data, then that's interesting. And so what I did is that then I looked at these preserved modules even further. And so the one way I analyzed the modules that were preserved was by looking at intramodular hubs. And so first, I'll just show you this little graph here. And what this is supposed to illustrate is I defined intramodular hubs by looking at a measure called eigengene correlation. And all that is the module eigengene is basically a measurement that's supposed to basically summarize the movement of the entire module. So a module consists of gene expression data for all the genes within the module. The eigengene is sort of supposed to summarize that. So genes that are highly correlated with a module eigengene are likely to be highly connected to other genes within that module because their movement is kind of indicative of the module as a whole. And so by looking at genes that were highly correlated with the module eigengene, I came up with a list of intramodular hubs, or highly connected genes, within these preserved modules. And so already, when you take this list and you look at which genes are differentially expressed from a patient with a normal liver to a fatty liver, or which genes are differentially methylated from a normal liver to a fatty liver, you're already seeing some very interesting genes. And then when you look at the functions of these genes, you actually see that in many ways, they're related to functions that previous scientists have associated with the progression of fatty liver disease, but they haven't necessarily pinpointed these specific genes. So that all of a sudden made it interesting. We all had all these candidate genes that related to known functions like inflammation or endoplasmic reticular stress or and other functions previously related to the progression of fatty liver disease, and now we've identified potentially highly connected, very significant genes, many of which were not identified before. So that was one way I analyzed the preserved modules. And the other way I wanted to analyze the preserved modules is by putting them in some actual biological context. Because as I mentioned, the networks I was constructing were entirely data-driven. They were unbiased. So I was constructing them, I was taking correlation from gene expression data without a care in the world for what those genes actually meant and what previous scientists had actually found out about them. So in order to get some biological context, I interfaced my data-driven network with a protein-protein interaction network. And on a protein-protein interaction network, each edge in a protein-protein interaction network is defined between two genes or the products of two genes where some scientist in the past has already discovered a known way in which these two genes interact. So if there's an edge connecting two genes in this network, that means that they are empirically related. And so once I was able to I took the nearest neighbors of my intramodular hubs and was able to reconstruct this protein-protein interaction network, then I was able to use this algorithm called the ENCODE algorithm to identify highly connected subclusters within this interaction network. And so if you have a highly connected subnetwork and you know that each edge in that subnetwork corresponds to a known biological relation, it's pretty likely that the genes in that subnetwork are related and potentially part of the same pathway or related to the same function. And so once I extracted those highly connected subclusters, which would ideally correspond to a biological function, I also wanted to look at how DNA methylation might affect that particular pathway or that function in question. And so to do that, I kind of annotated each edge and each node on this network. So a red node is a gene that's differentially expressed, and a yellow node is a gene red stands for one that's both differentially expressed and differentially methylated, and yellow stands for one that's just differentially expressed. And I also annotated each edge. So a red edge would correspond to a relationship between two genes where the DNA methylation profiles of those two genes grow more coordinated as you progress from the normal patients to the NAFLD patients to stage 3, the NASH patients. And so to do that, you have something called canonical correlation, which basically says how the variation in the DNA methylation profiles along the sequence of one gene explains the variation along the sequence of another gene. And from that measure, I was not only able to identify highly connected subnetworks, but as you can see, all these red edges correspond to two genes which are not only biologically related, but also the methylation profiles of those two genes are growing significantly more coordinated as disease progresses. And so there are a lot of interesting conclusions we can draw from that. So for instance, as you can see here, there are mostly all red edges in this network except for the edge connecting the two red genes. And so when I looked into that further, it turns out that those two genes that aren't connected by a red edge, so in other words, their methylation profiles don't grow more coordinated, they grow more disparate, or there's less of a connection between them, actually have antagonistic functions. So one's function is supposed to lead to more of this molecule called a serine threonine kinase, and the other gene is supposed to regulate the production of that molecule and lead to less of that molecule. And so, as you can see, what this was scientists with the facilities to actually perturb this network might look at this and say, oh, well, that's interesting. Maybe these two genes, if one becomes highly dysregulated relative to the other, that might affect the balance in this crucial function, and as a result, it might be affecting disease progression. And so, in cluster 2, as you can see here, so this cluster related to the function associated with this cluster was related to folic acid metabolism, and that was another new function which people had previously associated with. People had previously linked folic acid to as a facilitator of cell division and cell growth, and potentially but they hadn't linked it to fatty liver disease, and so potentially folic acid metabolism, should that function be dysregulated, could prove to be a link between developing fatty liver disease and then the cell division and cell proliferation that characterizes cancer down the line. And then in cluster 3, the function associated with this cluster was related to endoplasmic reticular stress. And the endoplasmic reticulum, or the ER, is supposed to break down proteins or process proteins in your cell. And so when it gets stressed or when there's an overload of proteins, that can also lead to inflammation that characterizes NASH. And so we identified several new genes or we've proposed new ways in which endoplasmic reticular stress can occur, and we've proposed a way in which DNA methylation and increased coordination in DNA methylation might be driving dysregulation in this particular function. So that was sort of what I did for my Intel project, and then I'll go into, I guess, for a couple of minutes what I'm currently doing, and then I'll open it up to questions. So what I'm currently doing is looking for different pathological differences between various stages of the disease. So as I mentioned, you go from a normal liver to a diseased liver to a NASH liver. Stage 1, stage 2, stage 3. And you can do something, you can cluster each sample. And so what that means is that you can basically construct a tree of sorts, kind of like a family tree, where at the bottom level, the genes that are, the samples that are connected are very related to each other, and then you progress to the upper level. When I clustered these samples according to all the data I had, they separated, group 1, group 2, group 3. But when I clustered these samples based on data from just one particular pathway or a couple particular pathways related to, for instance, cancer, then what was really interesting is you can see that the NASH samples separated into two distinct groups. And because some people who have NASH will eventually go on to develop liver cancer, whereas others who have NASH will just stay at NASH, the fact that these two groups have separated and have remained distinct when you cluster according to data from a number of different pathways was pretty interesting, and would indicate that there might be that might be what's underlying the progression from NASH to cancer, since we're clustering based on data from pathways related to cancer. And then so the second point of interest that we're currently looking at is that we have a way of measuring how dysregulated certain pathways are. So you fit a curve based on data from normal samples, and based on how the disease samples deviate from that curve for data from a given pathway, you can measure how dysregulated they are. So as you can see, that's called a pathway dysregulation score, or a PDS. And so as you can see, when you look at the diagram on the left, which corresponds to functions we associate with fat metabolism and functions we typically associate with the onset of NAFLD, the NASH samples don't really separate. But then when you move on and quantify dysregulation based on data from pathways related to cancer, again, you see this division of the NASH samples. And then so you can look at, in order to examine why some patients with NASH might progress to further stages of disease while others won't, you can again look at module preservation from the normal condition to then the first group of NASH patients versus the second group of NASH patients. And what we found is that the modules were actually more preserved going from the normal patients to the first group of NASH patients than they were when you're going from the normal patients to the second group of NASH patients. And so that would indicate again that the second group of NASH patients are more likely to progress to liver cancer. And so now what you would do with that information was that you would then, of course, look for more biological context. And then that would be what we would then do in the future. And then potentially these results could lead to a more complete understanding of why some individuals progress to liver cancer and others just stay at more benign forms of the disease. So a couple conclusions. As I mentioned, genes work in teams and networks, and you have to analyze them that way. It's only fair. And so this network-based approach revealed new insights and subtle trends that others previously missed because they were only looking at one gene at a time and whether or not it was differentially expressed or differentially methylated. And then these functional differences that you're seeing between these two subdivisions of NASH patients may in the future yield valuable insights for scientists when we interpret them and when we impose some biological context to them. And so with that, thank you, and I guess I'll open it up to questions.
Okay, we only have time for a few questions, so I'll just take the most popular ones. If the Boltzmann theorem of computational clusters is assumed true, how could pathological differences be modeled?
No idea, but whoever asked that question knows more about that theorem than I do.
Okay, if liver cancer is an assumed Nash equilibrium, which GO pathway is dysregulated?
Repeat that question.
If liver cancer is an assumed Nash equilibrium, which GO pathway is dysregulated?
Again, no idea, but I didn't really look at, yeah, next question.
Okay, how much did the professors you worked with contribute to your project?
So I was working remotely with with a couple people at UC San Diego, and so we came up with the idea together, and that was it was cool because we were able to come up with an independent project as opposed to most of the times when you're working on an offshoot of an ongoing project. And so I was responsible for doing all the analysis, for proposing a lot of the methods we used, and then what would happen was that I would check in via conference call or just a call or email about once or twice a week. So it was, I think it was mostly independently driven, and I think I learned a lot because of that.
Okay. What classes did you take to learn about bioinformatics?
So actually, it's more a question of what classes I didn't take, and I didn't take very many computer science classes at Harker, which is something I regret, and I would recommend that all of you underclassmen do. But the classes that did help me were my AP Biology class. That was tremendously valuable in understanding DNA methylation, and that was kind of what got me interested in bioinformatics in the first place. And then my math classes, a lot of the math I used was related to the linear algebra, and all those classes are offered by Harker. So linear algebra and AP Bio, I'd say, 2 classes.
Okay, I think we're out of time. So just one question from your friends back there. On a scale of 1 to 10, how happy is a happy liver?
An 11.
Okay, thank you.
All right, thank you.